So this is just going to be somewhat brief of an overview of the group of organisms referred to as the Eubacteria, or true bacteria, which is a whole domain of life. And I'm going to try to condense it down here into just one short lecture as we look at diversity of organisms. And this also kind of follows our progress of life through probiont cells to the first cells and multi-cell, and then when we look at diversity of life, we're coming back to those kind of first cells, which would be bacterial cells, and another group which we'll talk about separately, the other domain of archaea. And in that lecture I'll compare the archaea to the true bacteria. There's a whole microbiology course. The whole semester covers bacteria, structures within the bacteria, their physiology, and a whole variety of other aspects of their biology. So that's a whole semester, and I'm just going to boil it down here into just one short little talk. So what sort of characteristics do we have? For organisms in this group, obviously, one of the first things that always comes up is we say they have no nucleus. What does that mean? It means essentially their genetic material is free in the cytoplasm. And it's free, meaning it's not contained within a membrane. Which then kind of expands on this. It's not that they do not only have a nucleus, that the nucleus is missing and they have other sorts of membrane structures. There's no internal membranes. And so the nucleus is a membrane structure, and they don't have any of those internal membranes, at least not the way eukaryotic cells do. They can have some membranous little compartments in some of them, and they can have other cytoplasmic structures. So one of the misconceptions is that a bacteria is this little sac that's just full of liquid, and it has its DNA, and then that's it. It's empty because there's no organelles and there's no membranes. But that's not really true. There are all sorts of ways that material is stored within the cytoplasm of a bacteria, in little granules, for example. And little structures they can also kind of form, little pockets called mesosomes, which are kind of like membranous structures, like a little organelle. And things called inclusions, which are, again, groupings of molecules, like the granules, all together in one compartment. So they can alter somewhat the concentration of things in the cytoplasm by clustering or grouping things together. But the main thing we should be focused on is that the genetic material is not surrounded by a membrane. In addition to that, they have an organelle that we do as well, and it's in the cytoplasm here, and those are the ribosomes. So ribosomes, remember, are structures composed of RNA and protein, and they're used to make protein. So they're the structures used to carry out the process of translation, where RNA information is read, and from that amino acids are assembled, and we build new polypeptides. We do that in our cytoplasm. Bacteria do that in their cytoplasm, and they have ribosomes, and we have ribosomes, and they are slightly different, but they're very similar, and they work pretty much the same way. So those are some similarities. Now, their genetic material is DNA, and it is DNA structurally, essentially like our DNA with nucleotides, A's, T's, G's, C's. The individual nucleotide structures are the same. The way they're put together is the same. All the rules essentially are the same for copying them or reading them and so forth. One of the main difference, though, here, as you can see in my little drawing here, is that this DNA is circular. It's circular DNA. So circular DNA means that there are no ends to it. So our DNA has an end. And one of the problems with having an end is in the replication process. So once you go through that, you'll find that every time it's replicated, there's a little bit on the end of each chromosome, or one strand of each chromosome, that can't be copied. And so it's lost. And so the chromosomes shorten over time, Whereas because these chromosomes are circular, that doesn't happen to them, so all of them can be replicated. So that's an advantage that these cells have that our cells do not have. So the cytosol here, that's the liquid part of what we call the cytoplasm. Cytoplasm is this whole region with all the structures within it and the liquid within it. It's water and all kinds of dissolved molecules, and then all these other structures that we find in it, which some of them are similar. This is where they're going to carry out many of their chemical reactions, like glycolysis. Unlike our cells, we carry out the citric acid cycle inside the mitochondria, but they carry out the citric acid cycle in the cytoplasm because they don't have mitochondria. I'll talk about metabolism. Now, in our cells, we have mitochondria that also have an electron transport chain that are used to take glucose and break it down, and then take electrons and then carry on those electrons for energy, and then use them to make ATP. Well, bacteria cells can do the exact same thing. This is part of the serial endosymbiosis and the origin of the mitochondria. All right, so they also have an electron transport chain. Okay, but that electron transport chain is located here in the cell membrane. So what I'm going to do is now kind of blow up an area, here. And so what we have is kind of have this is the cytosol. This is the cell membrane, which is pretty much just like our cell membrane with phospholipids, phospholipid bilayer, phospholipids. Like this, with proteins then, embedded in it and through it and so on. And so what they have here is then a cell, a cell wall. That's what I'm looking at here. Yeah. So the cell wall of bacteria is unique. Well, now we don't have cell walls. Animals don't generally, but plants do. But they're totally different. A plant cell wall and a bacterial cell wall, not the same thing, at all. Similar function, but completely different material. So their cell wall is made up of a chemical called peptidoglycan. Which, if you break down the name, glycan means sugar. Peptid refers to like a peptide or polypeptide, which is a group of amino acids, and that's essentially what it is. It's a bunch of sugar molecules like this that are linked in chains, and then there are many of these, and then they're linked together. So a chain of sugars, a chain of sugars, a chain of sugars, and then every so often there are little bridges where peptides kind of link them together, and it starts to form this like mesh, like structure that is the peptidoglycan structure. And so that's what we find here, and then there are different thicknesses of this peptidoglycan. So some groups of bacteria have a very thick cell wall, and we refer to them as gram positive. And they have the thick peptidoglycan. So it's many layers of these sugars all put together. There are also another main group of bacteria that we call gram negative. And for them, this peptidoglycan structure is very thin. So instead of being a really thick structure, it's a really thin structure with only a few layers of the sugars. And they have other things that go beyond that. Then they have like a second phospholipid bilayer that go outside that, and there's some other variations to this, which again is kind of beyond our scope, but we're kind of getting into that. Now, I put a space here actually. So this is the cytoplasm. This is the membrane. Then there's a space, and there's a cell wall. So this space is important. Okay, it's a space called the periplasm. So the cytosol makes up the cytoplasm. So cytoplasm is one space, periplasm is a second space. And why that's significant is, I was talking about the electron transport chain here when I started this, and that's because the electron transport chain are proteins, they're transmembrane proteins. They carry electrons, they're pumps that they pump hydrogen ions or protons into a space. And then eventually they're used to make ATP in the cell, or in the mitochondria. Our cells do it, but here in the bacteria, it's the same sort of thing. They have the same sorts of proteins. They're embedded in a membrane. They pump protons across the membrane. There's a structure separate from this, not the actual pumps here, but an FOF1 ATPase that allows diffusion of them, and that energy is used to make ATP. So essentially what happens in our mitochondria happens in the bacterial cell, and it's structured, and the process of that is just about the same, but the locations are a little bit different. It's just that in a bacteria, because there are no internal membranes and there's no mitochondria, it just happens within the outer cell membrane itself, and then there's a second space. So again, a bacteria isn't just a little bag with just liquid inside it and nothing much else. There's actually more structure to it. Okay, so there's actually a layered structure, and there's a secondary space called the periplasm. So that is important because it allows you to see that they're a little more complex than you may have thought they were, and they can do then more complex things than you thought maybe the bacteria can do. All right, so we have a cell membrane like ours, a cell wall that is different, unique, but this structure is called peptidoglycan. Different kinds of bacteria have different thicknesses of that layer, and their cell walls then generally have a lot of other characteristics, which we can't get into, with different chemical compositions and other sorts of things that make them different or unique, and they're put into two groups referred to as gram-positive bacteria and gram-negative bacteria. They're the biggest ones. And they can have some other structures in their cytoplasm besides the chromosome, which is a circular chromosome. They can have ribosomes, and they can have all kinds of other little things. In addition to that, because we're talking about the DNA, they can also have plasmids. Which is significant. So this is extra chromosomal DNA. And it's also in a circular structure in a loop. So it's genetic material held separate from the chromosome. Still in the cell, still within the cytoplasm of the cell, still structured just like typical DNA, but it's separate, right? And they're small, and then these structures can potentially be passed on to other bacterial cells. So this is another part now of the structure of bacteria, that most bacteria will have structures on the outside of them, like this, called pili. And these little extensions then allow them to connect to other cells, so sometimes to other bacterial cells or to other cells of other organisms, like our cells, for example, or help them move around. Even there's some of them they can help them do that, almost like crawl with them. But in the case that what I am discussing sort of with you is that the pili can extend in a particular kind of structure. So there's different kinds of pili, then they have different kinds of jobs. One of them just doesn't do everything. There's a specific type of one that is coded for in one of these plasmids, and it can then create a special pilus that can grow, see, like this, and then connect one cell. This is one bacterial cell, bacteria A, this is bacteria B, and then this bacteria here can have a plasmid. It can then make a copy of that plasmid, and then it can take one of those copies and push it through to the other cell. So this cell here can then get new genetic material. So bacteria can transfer genetic material to other bacteria. This is one of the ways, say, antibiotic resistance is gained, where a bacteria doesn't have antibiotic resistance, but then this one does. It's coded for in this plasmid, and then they can pass that on to other cells, giving them that resistance, and a whole variety of other things, not just antibiotic resistance, but that's how genetic information can be passed on, and they can become then more complex. These things are typically separate from the chromosomal DNA, but there are ways that they can actually become integrated in other sorts of processes. So bacteria now have structures also called pili that allow them to do other sorts of things, and then some bacteria have flagella, different from pili. Now, the significance of flagella is they're made up of a specific protein called flagellin, and then they have certain motor proteins that spin. Okay, so essentially when these structures are embedded, you have a flagella like this, it's connected then to these motor proteins that form these disc-like structures and that are embedded in cell wall and cell membrane, and then these structures will actually spin and rotate, and that causes this whole flagella to spin and rotate. The purpose of talking about that is that it's very different than eukaryotic flagella. So some of our cells have flagella, and they have structures called cilia. And the cilia and flagella are exactly alike in terms of their internal structure. We'll get to them as we start to talk about protists and mechanisms of movement as well. The flagella of bacteria, very different, completely different. Proteins make them up. The way in which they're structured is different, and the way in which they move is completely different. So the bacterial flagella is, once again, a totally unique structure that's very different than what we see in our eukaryotic cells. The way their cell wall is very different from eukaryotic cell walls. Plant cell walls are made up of different material. They're not made up of peptidoglycans. So kind of keep some of those things together because they are unique. And I'm trying to kind of point out things that are very unique that they have, and there are a few things also that overlap, and they're essentially the same. So the glycocalyx is now something else that most bacteria have in one form or another, and that is essentially outside now of the cell wall is this sugar coating. Typically referred to as a glycocalyx. So it can be organized into a variety of different ways. It could be highly organized into a capsule. It could be completely disorganized into something called a slime coat. It could be used for a variety of different things. Sometimes it protects the bacteria. Sometimes it helps the bacteria stick to certain things. So what exact all the details is also a far more complex topic than what we're covering, but just know that outside of the cell wall, they typically have other layers. Some of them even have protein layers called an S coat that kind of protects them as well. So there's more and more levels of complexity to this than just the most sort of simple sort of thing. I already mentioned plasmids and the ribosomes. So again, there's a lot more of details that we could talk about in terms of structure. The last thing I'll mention about structure is that bacterial cells shape have a whole variety of different shapes. Generally, you have round, perfectly round spheres called cocci. You have rod-shaped cells called bacilli. There are these sort of twisty, they're actually kind of corkscrew, kind of called spirelli. There are these comma kind of shaped ones like this, called vibrio. And there's others as well, but these are sort of the most common ones, and really most of the time in, say, a microbiology class, you're typically working with cocci and bacilli, just really these two types, and then we discuss these others. A lot of the vibrios are pathogenic, and the spirelli have unique things they do with their flagella, where it's actually in the periplasm and it makes them move around. So there's some variations that they have of some of the same structures. So just so that you know. Now, metabolism is something I'll also spend a minute or two talking about, and then we'll talk about their ecological roles. And this will be kind of some of the structure that we're going to get into with as we go through diversity of most living things. I'm going to talk about form and function, a lot of structures, and then the function of those structures, and then eventually we'll get into the role of that group in the environment or to ecosystems or as predators or prey, variety of those sorts of things that we'll get into as we think everything will sort of be structured that same sort of way. So as far as metabolism, again, they have a lot of the same metabolism that we do. They run glycolysis the same as us. So same enzymes, same intermediate molecules, same process, everything pretty much the same. Glycolysis is a process that can occur with or without oxygen. So we don't need oxygen to do it, and neither do bacteria. We need oxygen, though, to continue on beyond glycolysis and do other sorts of things. So we run pretty much just aerobic respiration. Bacteria, not all of them, no, but some can do anaerobic respiration. So without oxygen, some of them will run fermentation. So not all bacteria do fermentation, not all bacteria do it the same way. Some of them have different end products from fermentation. So sometimes alcohols are produced, acids are produced, a variety of different types of molecules can be produced from fermentation. But the purpose of this is so that glycolysis can essentially continue to run. So without fermentation, if you're familiar with glycolysis, they're NAD plus molecules that pick up electrons, and they become NADHs, and those NADHs go on to the electron transport chain. If there's no electron transport chain, then they don't have anywhere to go, which means eventually those NADs run out, and even glycolysis will stop. So even though it doesn't require oxygen, it'll still stop in the absence of oxygen, unless there's some way of getting all the things it needs to continue to run, and that's what fermentation does. In addition to that, bacteria have a variety of metabolic variations that can make them unique to make a whole variety of different types of products. And so unique metabolism of bacteria can make essentially you and a giant sequoia actually more alike than, say, these two bacterial cells. So what I'm saying is two bacterial cells can be more genetically different from one another than you are different from a tree. So you and the tree are much more similar than two different types of bacteria might be to one another. Because in addition to all sorts of variety of different structures that you're starting to just get a little bit of an idea that they can have or not have. Some have a lot of the pili, some don't. Some have other things called fimbria and some don't. Some have flagella and some don't. Some have the flagella in different arrangements and some don't. Some have different types of cell walls. Some have different sorts of layers. And then within them physiologically, there's a whole entire variety of different types of metabolism that can occur that we don't do anything like. That it's very unique, and different groups do certain things, again, that we don't, and they do things different than other groups. And so there's a lot more metabolic diversity than we typically see in eukaryotes, right? So there is actually a lot of diversity in both process and products. And those can be good things, they can be beneficial, they can be used by humans in manufacturing. One of the things that we actually use bacteria for. They can produce the waste products, they can produce toxins. It could be things that make them very dangerous, as well. So again, all sorts of things, being good things, bad things, neutral things, and then that kind of brings us to their roles. So what do bacteria actually do? What are their roles in the world? And again, a whole variety of different roles. Some of the first photosynthetic organisms were bacteria that produced oxygen for the atmosphere, and they still do. So there's still bacteria that are photosynthetic today, that run photosynthesis and take carbon dioxide from the atmosphere and put oxygen into the atmosphere. A lot of bacteria are involved in nutrient cycling. in the environment. So breaking down certain nutrients and turning them into other sorts of molecules that can then be used by other organisms. So decomposing of organisms with fungi, bacteria play that role as well. Bacteria are food to a variety of organisms. There are organisms that eat bacteria and eat bacterial films. So bacteria are the main primary nutrient for a whole variety of other sorts of things. Many bacteria live symbiotically within and on the surfaces of a variety of organisms. So we have what's called a microbiome. So we have bacteria that live on us and bacteria that live inside us. The bacteria carry out a variety of chemical reactions actually that we can't do. And if you would remove those bacteria, you would be harmed because you would be lacking in the ability to digest and process certain things. You'd be missing certain vitamins because they actually make them or allow you to be able to absorb them, and you wouldn't be able to without them. And they do a whole variety of other things that are still being studied, that people are trying to understand. Many diseases that we have as humans, we find are not caused by bacteria, like we're thinking about pathogens, but are prevented by the bacteria that we have. And if we lose those bacteria, we are much more likely to have that disease. They actually protect us from getting a variety of things. We typically know of bacteria as germs, people call them, so they cause disease. And that's the way they have been studied mostly for a very long time. And so there are a variety of bacterial pathogens. Some have the outer layer of the organism has chemicals that are toxic in some way to a host organism and make the organism ill. Others can secrete or release chemicals that make the host very ill. But generally, we have a lot of bacteria within us. We actually have more bacterial genes than human genes operating in our body at any one time. And they are all beneficial, helping us overall. And they do many other organisms, both plants and all sorts of other animals. Symbiotic relationships with bacteria are beneficial. So they have ecological roles that are beneficial. They have ecological roles, or they have symbiotic roles with a variety of organisms. They can be nutrients to other organisms. They can cycle nutrients, and then they can also be disease-causing organisms as well. So there's a whole lot to study when studying bacteria. And the main point here is to talk about, so what is this group of prokaryotes? What are some of their key characteristics that make them unique on their own? What are things that we share that are somewhat similar? And then what are things that are different? Okay, so similar membrane to our membrane. The ribosomes, slightly different, but basically the same and carry out the same thing. The structure of the DNA itself, basically the same, but its organization is different. It's also not packed into a nucleus, which is different. The process of transcription and translation, they're pretty much the same as far as they occur. Processes like glycolysis and the citric acid cycle, pretty much the same. As we go outside the cell and start to look at structure, then they get very different. So having the cell wall and having all these unique kind of flagella, the structure called pili and fimbria, which I didn't write down up here, but it's more of a specific bacterial structure. We don't have that. So make sure you're kind of familiar with at least these terms and the basic things that I talked about and outlined up here as far as what to know about bacteria. Some of this will be repeated a little bit as I talk about the Archaea, which is the other domain of prokaryotes, because I'll be doing a comparison between the Archaea and the bacteria.